This lecture is on liver anatomy and physiology. It's chapter 9 in your Hagen Answered. So the liver occupies most of the right upper quadrant. It's going to occupy all of the right hypochondrium, most of the epigastrium, and some of the left hypochondrium as well. It's going to be inferior to the diaphragm. Most of the right lobes is protected by the ribs, and in the epigastric region, the liver is going to extend several centimeters below the xiphoid process and also to the left of the midline. It's going to be anterior and medial to the fundus of the stomach, and it is superior to the right kidney. Let's talk about the lobes. You'll notice that there are 4 bullet points for lobes of the liver, even though you've learned there are 3 lobes. So the first lobe is the right lobe of the liver, and we're going to turn on our laser pointer here. There we go. Right lobe of the liver, right here. Okay, and when we're looking at it from underneath, this is the right lobe of the liver here. Gallbladder fundus is anterior. IVC is posterior, just to orient you to the screen. Okay, then we've got the left lobe of the liver, right there, down here. Here's our left lobe here. Okay, we have the caudate lobe. The caudate lobe sits at the back of the liver, so we can't see it in this picture. Caudate lobe is here between the left lobe and the IVC. Now, we talked about there's a 4th bullet point. So the 4th bullet point is for this lobe right here, which is the medial left lobe of the liver, or also known as the quadrate lobe. So you got the right lobe, the left lobe, the caudate lobe, and the quadrate lobe, also known as the medial left lobe. So before I give you the answers, we're going to test our anatomy knowledge when it comes to the liver. So I want you to go ahead and pause this, take a moment, see if you can name all of these items, and then we'll go over them together. Okay, let's check our understanding and see how you did. We're going to start right up here in this corner right here. So this is pointing to this triangular little thing right here. This is the left triangular ligament. Underneath that, right here, we've got the left lobe. The next one is this big ligament here that attaches the liver to the front of the abdominal wall, and that's the falciform ligament. At the end of the terminal end of the falciform ligament, you're going to find the ligamentum teres. Sonographically, you're going to see this as a bright triangular or circular focus in the left liver. All right, this one here is pointing to the inferior margin of the right lobe. Under here, we got this little green guy. That's the fundus of the gallbladder. This box right here is pointing to the right lobe of the liver. This box here is pointing to the costal surface of the liver. Okay, the costal surface, which is the part that sits up against the ribs. And then we have the right triangular ligament here, the diaphragm right up here, and these two here are the coronary ligaments. How did you do? So now we're going to try another one from the bottom side of the liver, or the inferior side. Go ahead and pause the recording, see how many of these you can get right. Let's see how you did. So we're going to start right up here at the top. This one is pointing to that medial left lobe, also known as the quadrate lobe. We already talked about that a little bit. The next one is pointing to these 2 red vessels right here. These 2 red vessels, as seen from underneath, are the hepatic arteries. Over here we have the left lobe of the liver. Left lobe of the liver. This blue one, this blue vessel, is the portal vein. Then down here we've got the caudate lobe sitting between the left lobe and the IVC. The blue vessel is the IVC. Over here, this small section posterior to the posterior side of the liver is the bare area. That's a section of the liver that meets up against the diaphragm and is not covered by Glissen's capsule. The rest of the liver is covered by Glissen's capsule except for this small area here called the bare area. And this is how it looks as seen from the underside of the liver. Over here, we've got the right lobe. Then this box right here, we're pointing to the cystic duct. And this box right here is pointing to the hepatic duct. This central section right here contains the portal triad, the main portal vein, the hepatic arteries, and the hepatic duct / common bile duct right here at the porta hepatis. We can also divide the liver into segments using Quino's system of hepatic nomenclature, and it divides the liver into 8 segments. The first segment is the caudate lobe. Remember, it's posterior. The second and third segments are the left lateral segments. The superior lateral is number 2. The inferior lateral is number 3. The left hepatic vein is going to feed these and sit between those and the medial part of the left lobe, which is 4A and 4B. The left portal vein is going to bisect and run between sections 2 and segments 2 and 3. Now, 4A and 4B are also the medial left lobe, also known as the quadrate lobe. 4A is the superior medial left lobe, and 4B is the inferior medial left lobe. Then next to that, we're going to have the right lobe. 5, 6, 7, and 8 are going to start down in the bottom medial, and they're going to go clockwise. So 5 is medial and inferior and anterior. And then 6 is right next to it, lateral, also posterior, and that's going to be inferior as well. Superior to that is going to be number 7, which is the superior posterior segment of the right lobe. And then segment 8, is the superior anterior segment of the right lobe. Again, that right hepatic vein is going to run through sections 6 and 7. 5 and 8 are going to be the right anterior portal vein that bisects those. Right posterior portal vein is going to bisect 6 and 7 again. Ligaments and fissures that you need to be aware of, that you need to understand where they're at when you're talking about the anatomy of the liver. These things are going to be bright echoes on ultrasound. They're usually linear, so they're going to be specular reflectors, which is why they have such a strong reflection. Glissen's capsule is the surrounding or the covering of the liver. The main lobar fissure, this is the one that's going to be an echogenic line between the gallbladder and the portal vein. The falciform ligament is going to separate the left lobe from the right lobe. The ligamentum teres is going to be the terminal end of the falciform ligament. Remember, we said it's going to be a circular or triangular hyperechoic focus in the left lobe inferior. And then the ligamentum venosum. The ligamentum venosum is the remnants of the umbilical vein. We no longer need it once we're born. It's part of the fetal circulation, and it's going to separate the caudate lobe from the left lobe. It'll be a bright linear echo between the caudate lobe and the left lobe of the liver. We're going to take a quick look at as many of those ligaments and fissures as we can. Here we have the portal vein, the gallbladder, and that main lobar fissure, which is going to run straight from the portal vein over to the gallbladder. In this picture here, we got that ligamentum teres. Remember, we said it was going to be a hyperechoic, circular, or triangular focus in the left lobe of the liver, which is right here. Okay. Over here, we've got that ligamentum venosum right here. That's what the arrow is pointing to. Left lobe of the liver here. Caudate lobe of the liver squeezed between the left lobe and the IVC. All right, now the vascular system. The vasculature of the portal venous system, we're going to have the main portal vein. It's going to split into the right portal vein, feed the right side of the liver, and the left portal vein. You're going to have extra branches on people. There's a lot of anatomic variation. Think of it as a tree with the main portal vein as the trunk, and the right anterior, right posterior, and left portal veins as the 3 main branches, and then lots of little branches coming off of those. It's going to carry blood from the bowel to the liver. It's one of the main sources of blood for the liver, and it does carry deoxygenated blood to the liver, but it still carries enough oxygen in that deoxygenated vein to feed the liver. So remember that the portal venous system enters the liver from inferior. We looked at the portal triad and the porta hepatis on the inferior surface of the liver, and it's going to run vertically into the liver, branch out into many, many branches, and feed all parts of the liver. Now, at the top of the liver, we also have a drainage system because the blood has to drain from the liver as well as go into the liver. And the system that drains the liver is the hepatic venous system. It also has a number of little branches that come together. They're like tributaries, come together to form larger tributaries. So it's sort of like mountain streams coming together to form creeks and then rivers, and then one big river like the Yellowstone. Okay, so if you think of it that way, it's an upside down tree. All those little branches come together. They're going to merge into either the right hepatic vein, the middle hepatic vein, or the left hepatic vein. And then those hepatic veins are going to drain blood from the liver into the IVC, superior and posterior, at the top and the back. It's towards the back. And all of that is then going to go into the IVC and to the right atrium of the heart. The hepatic veins have less echogenic borders than the portal veins because of their histology and the organization of their of the tissues in the or the cells in the walls. So they're going to be less echogenic than the portal veins. That's one of the ways that you can tell whether you're looking at a portal vein or a hepatic vein. Sonographically, you're going to see those hepatic veins dump into the IVC at the superior posterior part of the liver. And what you're looking for is the bunny or the moose head sign. So here we got a nice Playboy bunny. We've talked about the Playboy bunny and everybody having one. But some people don't have a Playboy bunny. They either have a deer or a moose head. So you'll see an elongated IVC and then several branches of the hepatic veins that dump into that IVC. Here I just wanted to show you that in transverse, the difference between the echogenicity of the portal veins versus the hepatic veins, and also where they run in relationship to the IVC. So in both of these pictures, we have the IVC at the posterior section of the liver. Notice that the hepatic veins connect to the IVC. The portal veins run anterior to the IVC and never connect with the IVC. The portal venous system does not ever connect to the IVC. You do want to remember that. It's always going to run anterior to it. Now, notice that the portal vein walls are nice and bright. Over here on the hepatic side, we do have some bright spots. But as a general rule, the walls of the hepatic veins are not as bright as the portal veins. I've always liked this slide because it gives you an excellent visualization of what the portal venous system and the hepatic venous system are doing in there together. That portal venous system is coming from inferior to superior and branching out like a tree. The hepatic venous system is all intertwined with it and is draining the blood superior and posterior and into the IVC. Now coming into the porta hepatis, you're going to have the portal vein and the hepatic artery entering. You're going to have the bile duct exiting. They're called the portal triad because they accompany each other. Wherever you see a portal vein, no matter how small the branch, you're going to find a bile duct and a hepatic artery to go with it. So I just want to talk about that hepatic artery for just a moment. It's going to carry the oxygenated blood from the aorta to the liver. The portal vein, remember, supplies most of the blood to the liver, but the hepatic artery is an important secondary source, and it's going to carry the oxygenated blood because the portal vein has deoxygenated blood in it. So remember, it's going to start on the celiac trunk, which is the first anterior branch of the aorta, and it's going to be called the common hepatic artery as it comes off of the celiac trunk. The common hepatic artery is going to branch into the gastroduodenal artery and the proper hepatic artery. The proper hepatic artery is going to enter the porta hepatis. It's going to split just like the portal vein does into a right and a left hepatic artery. And yes, there is a right posterior hepatic artery following that right posterior portal vein. And then just like the portal vein, it's going to branch out. It's going to stick with its partner and run throughout the liver. So you may have heard about the Mickey Mouse sign. I'm not keen on that nomenclature, but I'm going to show you where it's at. Okay, so let's get our laser pointer out. Here's that portal vein right here. Anterior to that, you're going to see right here this tiny little dot. I'm going to move the laser pointer so you can see it. That's the common bile duct. Notice in this picture here, the hepatic artery does not form a second ear of the Mickey Mouse. It's stretched out. Okay, and this is why I don't like calling it the Mickey Mouse sign, because that's like textbook and very hard to get. And more often than not, you're going to see something more similar to this. Now over here, we do have that sort of Mickey Mouse sign. You're going to see the portal vein here, common bile duct here, hepatic arteries, this little tiny dot over here. So yes, they do form the ears and the portal vein forms the head. You got the main lobar fissure running up here to the gallbladder. Okay, and then back here you got the IVC. The functions, the primary functions of the liver, and the hepatic metabolic functions, the main ones are going to be converting dietary sugars into carbohydrates, and then, carbohydrates into glucose. So we're going to, the dietary sugars, the carbohydrates are going to be converted to glucose. The glucose gets converted to glycogen and then gets stored in the liver. And when glucose is needed, it breaks down the glycogen and releases glucose back into the blood. It's also a major site for metabolizing fats. The dietary fats are going to be converted in the hepatocytes to lipoproteins, the form in which fats are going to be transported throughout the body for storage or use, and the stored fats can be transported to the liver and converted into energy. The liver's also going to manufacture some plasma proteins. It's going to convert excess amino acids to fatty acids and urea. It's going to remove nutrients from the blood, and it's going to phagocytize bacteria and worn out red blood cells. It also has a role in digestion. It's going to secrete bile, which is important to the digestion of fats. Bilirubin is going to be a pigment that's released when the red blood cells are broken down, and that is also excreted in the bile. It has storage functions because it's going to store certain vitamins and also store iron. And then it has a detoxification function, which I'm sure all of you have heard about because it's all the rage right now. It's going to detoxify many drugs and poisons that enter the body, as well as waste products from the body's metabolic processes. So I want to talk a little bit more about bilirubin. This is going to be a breakdown product of the hemoglobin. Remember, we said it comes about when we break down red blood cells. The red blood cells have a life cycle of about 120 days, and then they become trapped and broken down primarily in the spleen. The hemoglobin gets released from the blood cells and is converted to bilirubin, and that floats around in our bloodstream. The bilirubin becomes attached to albumin, and that is then transported to the liver. The liver separates the bilirubin from the albumin in the hepatocytes, and it secretes it into the bowel via the biliary system. That biliary system is going to contain bile. It's an excretory product of the liver. It's formed continuously by those hepatocytes, and it collects in the bile caniculi adjacent to those cells, and it's transported to the gut via the bile ducts. The main function of the gallbladder is to store the bile until we're ready to secrete it. Principal components of the bile are going to be water, bile salts, and bile pigments, primarily that bilirubin. Other components include cholesterol, lecithin, and proteins, and the primary functions of the bile are to emulsify the intestinal fat. and removal of waste products excreted by the liver. So when we talk about disease and the liver, we have different kinds. We have hepatic disease versus obstructive disorders. So hepatocellular disease is going to be diffuse. It's going to be the liver cells, the hepatocytes, which are the immediate problem. And usually we're going to treat diffuse liver disease medically with supportive measures and drugs, versus obstructive disorders, which is when something becomes blocked. Most often it's going to be bile excretion is being blocked, and usually we have to treat that surgically rather than with supportive measures. So when we're talking about biliary obstruction, what's happening is the hepatocytes pick up the bilirubin, conjugate it with those glucuronide molecules, but they can't dispose of it because something is blocking the system. So then the conjugated form gets regurgitated into the bloodstream, and that resultant elevation of the direct acting results in an bilirubin elevation. Okay, of the direct acting bilirubin. The indirect can also rise a little bit in the presence of biliary obstruction, but the direct bilirubin is the one that's going to predominate. It's the one we're going to see go up the most. Now the tricky part is that the direct bilirubin is also the predominant form in hepatocellular disease. So whether we're talking obstruction or diffuse disease, the direct bilirubin is the one that's going to be elevated the most, okay? And the excretion of the bilirubin is affected when the hepatocytes become damaged, and therefore the diseased hepatocytes continue to take in and conjugate the bilirubin, but again, they're unable to excrete it. So just like with obstruction, that accumulated conjugated bilirubin gets regurgitated back into the bloodstream to float around our body. Liver function tests that you should be familiar with because you are going to see them on orders as indications for exam. You'll see something that says elevated LFTs, or they may actually pinpoint the particular liver function test, LFT, that they performed. So you're going to need to know about aspartate aminotransferase, also known as AST, alanine aminotransferase, ALT, lactic acid dehydrogenase, LDH, alkaline phosphatase, bilirubin, the indirect, the direct, and the total bilirubin, prothrombin time, which is known as PT, and then albumin and globulins. These are all going to start to make a little more sense as you start clinicals, but you do want to know what these ones are, and I would study the pathology part of the book so that you know which ones elevate in the presence of which disease. And here is a little chart to kind of help you with that. These are the primary ones, the hepatocellular diseases. Remember, these are your diffuse diseases. You're going to see the bilirubin go up. You're going to see the albumin go down. You should see the other three, the AST, the ALT, and the alkaline phosphatase, go up. Okay, with obstruction, you're going to have a severe increase in bilirubin. Your albumin is probably going to remain normal. It's not going to go up. And then you may see a mild increase in AST and ALT, but you should see a severe increase in the alkaline phosphatase. The remaining 22 slides in this presentation go through how to image the liver. So we want to talk about patient prep, which should be NPO for 6 to 8 hours. We don't want them to eat prior to their exam. Patient positioning, typically this is going to be supine, possibly with the head slightly elevated. For certain parts of the liver, you may find yourself rolling them left lateral decube to get a better image. Breathing is going to be very important. You may need your patient to take deep breaths and hold them in order to get good pictures of the liver. Instrumentation, we're going to want a curvilinear probe, and we're going to want to talk about probably using a 3.5 to 5 megahertz to do the liver. You're going to want pictures in sagittal. You're going to want pictures in transverse. You're going to want color Doppler on parts of the liver, specifically the portal veins and the hepatic veins. You want to measure the length of the liver. And once you've done all that, you are done. So I'm going to try and run through each of these slides fairly quickly because I am running out of time if I'm going to keep this under 30 minutes. We're going to start with the left lobe in sagittal. You want to you want to take images from lateral to medial. So you're going to start far left and work your way towards the IVC. And your landmarks are what's going to help you figure out where you're at. So you're going to begin under the xiphoid process, sweep from the porta hepatis to the left until you no longer see the liver, and then you want to take the following images. A left lobe lateral using the stomach or the gastroesophageal junction or the gastroduodenal junction as your landmarks. The next one is going to be medial with the aorta in the picture, and the last picture is going to be more medial even than the aorta, so to the right of the aorta, including the left lobe, the caudate lobe, and the IVC. It should look something like this. Okay, here's our first picture. Left lobe lateral. We can see, let me put on my laser pointer. There we go. We got that triangular left lobe. We've got some pancreas in this picture. And we've got that GE junction, gastroesophageal junction, looking like a target. We do not see aorta in this picture at all. This is your first image of the left lobe in sagittal. The next image should include that aorta. It's a mid left lobe. You got the aorta. You've got the left lobe of the liver. Again, we can see a little bit of that pancreatic head right here and a little bit of that superior mesenteric vein coming up to join the splenic vein to form the portosplenic confluence. The next picture should include that IVC. This is your most medial part of the left lobe. It's going to be medial left lobe. Remember, this is sections 4A and 4B if we're using Quino. And that ligamentum venosum, that nice bright linear structure separating the left lobe from the caudate lobe. You may see the left hepatic vein in there. And posterior to the caudate lobe, you should have the IVC running. That's the sagittal left lobe. Next, we're going to do the left lobe in transverse. We're going to rotate the transducer 90 degrees to the right. We're going to start at the bottom of the liver, sweep up through the top of the liver until we reach the heart. And starting at the top of the liver, we're going to take the following images. We want lateral or medial segments divided by the left hepatic vein. We want the left lobe with the left portal vein. And we want the caudate lobe with the IVC, aorta, and the left vein. So here we go. Okay, we've got the superior picture. We went all the way up to the heart. We've got the liver here, those hepatic veins dumping into the IVC. That's the superior. The mid left lobe should include this left portal vein right here. See the bright walls? That tells us it's not a hepatic vein, it's a portal vein. We got main portal vein here. We got right going off this way, left one bisecting those anterior and posterior parts of the lobe. Okay, and then we said caudate lobe, aorta, IVC, left lobe of the liver, and the ligamentum venosum should be in this picture as well. That's that bright linear focus between the left lobe of the liver and the caudate lobe. Now you're done with the left lobe, we're going to run right into the right lobe. The right lobe, we want to use either a subcostal or an intercostal approach. You're going to have to try them and see which one works the best. Have the patient take in a deep breath. See how that changes your image. Figure out where your image is going to be the best and realize that you might have to move to get different parts of the liver. You're going to sweep from the porta hepatis right to the hemidiaphragm and the pleural space. Remember, we are in sagittal right now. So starting medial and working lateral, we're going to take the following images. You want the right lobe anterior, including the gallbladder, the main lobar fissure, and the main portal vein. And then you want the right lobe lateral with the hemidiaphragm and the pleural space. And then you want the right lobe posterior with the right kidney interface. So you want to see Morrison's pouch and be able to compare the kidney to the liver and also clear the pouch of fluid. So let's take a look at those. Medial, right lobe medial. So we're working from the IVC side over to the lateral side of the body here. We've got the portal vein, the common bile duct running in front of the portal vein. We got the gallbladder. This little bright spot right here that's running between the gallbladder and the portal vein is the main lobar fissure. This is our first sagittal image. Our second sagittal image should include the right lobe of the liver, the diaphragm, the right hemidiaphragm, and you should see this mirror image artifact of the pleural space. In this picture, we also have portal vein and hepatic vein. We can see the portal vein walls are bright, the hepatic vein walls not so much. But the important part of this picture is you've got that hemidiaphragm and the pleural space. Next picture is going to be that right posterior, where you're going to have the right lobe of the liver, the kidney, and between them, this space right here, Morrison's pouch. Transverse. Transverse, we want to rotate the transducer 90 degrees to the right for the right lobe of the liver. We're going to start at the bottom and sweep superior till we reach the dome. You want the following images from superior to inferior. You need the right lobe with the mid and right hepatic veins. You need the right lobe at the level of the right portal vein, preferably somewhere between the anterior and posterior, but you should see at least one of them in your picture. And then you want the right lobe posterolateral margin with the right kidney and the gallbladder if you can get it. Let's take a look at those. So superior, all the way at the top, we want to see that right portal vein, or right hepatic vein, that's the important one. We got another little branch right here, probably the mid. We can see that diaphragm. Okay, this is the superior transverse right lobe of the liver. Next, we want a mid. We want to see that main portal vein branching into the right anterior and right posterior portal veins. The IVC may be in the picture here. And here we've got the diaphragm over here. We know we're at a mid because we can see that portal vein. And then an inferior. Right inferior should include the liver, the Morrison's pouch, the right kidney and transverse, and if you can get it, the right gallbladder. Or I'm sorry, the gallbladder should be in your picture as well. All right. That's all the images you need in grayscale. You also need some color Doppler. You should have color Doppler of the main portal vein. You should have color Doppler of the hepatic veins, and you should have pulse wave Doppler of the main portal vein. They're going to look like this. Here's our color Doppler, main portal vein here, right posterior portal vein here. This green one is the hepatic artery. Portal vein with pulsed wave Doppler. Here they've gone ahead and used their angle correct. You want to be parallel if you're going to do that. The only way to get a velocity is to use your angle correct. And then notice that our portal vein flow waveform is pretty steady. It's a steady state, stable venous waveform. There's not a lot of variation to it. Okay, next we want color Doppler of those hepatic veins. We got 3 of them right. We got the left, the middle, and the right hepatic veins dumping into the IVC. That's this big blue part right down here. And notice they separated this into Quenos segments for you as well, so you know what you're looking at. And last but not least, you want to measure the liver. So you want to have the kidney in your image. You want to have the Morrison's pouch in there, and you want to be able to see the liver from the inferior tip all the way to the diaphragm. There's 2 ways to do this. This slide presentation was set up to do it the old school way. When you're in lab, I will show you the new school way to do it. But for purposes of this slide presentation, and you will see this in clinicals as well, we're going to measure diagonally from the inferior tip. It would be straight. We'd use our calipers to the diaphragm. Okay, and that's going to give you a measurement of the liver. And that's it for anatomy, physiology, and sonographic imaging of the liver.